Introduction. Welcome to today's lecture on the various non-radioactive labeling methods and applications of Southern blotting in molecular biology. Southern blotting, named after its inventor Edward Southern, is a powerful technique used for the detection and analysis of specific DNA sequences. It involves the separation of DNA fragments based on their size and subsequent hybridization with complementary DNA probes. Over the years, Southern blotting has been instrumental in numerous applications such as gene copy number analysis, DNA methylation studies, and forensic analyses. In this lecture, we will explore different non-radioactive labeling methods and the diverse applications of Southern blotting in various fields of research. Let's delve into the fascinating world of Southern blotting. Non-radioactive labeling methods for Southern blotting, such as deoxygenin, DIG, and Biodex labeling, offer safety, convenience, and cost-effectiveness. These methods allow precise detection and analysis of specific DNA sequences, expanding the applicability of Southern blotting to a wider scientific audience. In situ hybridization, ISH, combines Southern blotting with histological analysis to detect specific DNA or RNA sequences in intact cells or tissues. ISH helps study gene expression, cell types, and genetic abnormalities in diseases, providing valuable insights in various fields. In summary, DNA methylation is an important epigenetic modification that regulates gene expression. Methylation-specific Southern blotting is a technique used to study DNA methylation patterns by converting unmethylated cytosines to uracils and comparing band patterns to determine methylation status. In this section, we explore long-range restriction mapping using multiple Southern blots. The technique involves cutting DNA with restriction enzymes, separating fragments on a gel, and transferring them onto a membrane for detection using DNA probes. Multiple blots are used to map extended regions accurately, aiding in genomic sequencing and gene identification. In conclusion, quantitative Southern blotting is a powerful technique used to measure gene copy number variations in the genome. It allows researchers to identify and study genetic abnormalities associated with human diseases and contributes to our understanding of human genetics. Array-based Southern blotting is a technique that combines Southern blotting with high-throughput technology. It allows for the simultaneous detection and analysis of thousands of DNA sequences, providing insights into gene expression, DNA methylation, and genomic variations. This technique has revolutionized genomics and transcriptomics research, leading to the discovery of novel genes and biomarkers associated with diseases. Chromatin immunoprecipitation, CHIP, coupled with Southern blotting is a technique used to study protein-DNA interactions on a genome-wide scale. It involves cross-linking proteins and DNA, immunoprecipitating the complexes, reversing the crosslinks, and using Southern blotting to detect specific DNA sequences. This technique has greatly contributed to our knowledge of transcriptional regulation, epigenetics, and chromatin structure. Comparative genomic hybridization, CGH, detects variations across the genome. Coupling CGH with Southern blotting allows identification of genetic imbalances causing disease. CGH uses labeled DNAs to detect gains or losses in DNA segments, providing higher resolution for specific regions. CGH Southern blotting has revealed imbalances in developmental disorders, mental illnesses, and cancer, improving diagnostics and personalized medicine. Two-dimensional, 2D, gel electrophoresis allows proteins to be separated based on their properties. Combined with Southern blotting, it helps study protein-DNA interactions. This technique has been pivotal for understanding transcriptional regulation, DNA repair, and chromatin dynamics. In summary, single-strand conformational polymorphism, SSCP, analysis, when combined with Southern blotting, is a powerful technique for identifying sequence variations in DNA. It involves denaturing DNA samples, electrophoresing them, transferring them onto a membrane, and detecting sequence variations using specific DNA probes. This technique has greatly contributed to our understanding of genetic diseases, familial mutations, and population genetics. Southern blotting has evolved over time with various modifications. Slot blotting simplifies the process by immobilizing DNA directly onto a membrane, useful for quantification. Pulsed field gel electrophoresis, PFGE, allows separation of large DNA fragments, which can then be detected using Southern blotting. Combining Southern blotting with PCR improves sensitivity and specificity. These variants expand the technique's applications and enhance research capabilities. Southern blotting has important applications in forensics and evolutionary biology. In forensics, it helps analyze DNA fingerprints for identification. In evolutionary biology, it studies genetic variation for understanding population relationships and adaptive evolution. Southern blotting remains a crucial tool in these fields. Southern blotting, a versatile technique in molecular biology research, offers a range of non-radioactive labeling methods and applications. It is used for genomics, epigenetics, and forensic sciences, allowing specific DNA detection and analysis. Additionally, it can be combined with other techniques for enhanced insights. By understanding its variants, advantages, and specific applications, researchers can utilize Southern blotting to study genetic diversity and contribute to genetic profiling and evolutionary biology research. The technique continues to be essential, helping unravel DNA complexities and uncover the mysteries of life.